You guys are about to listen to a haiku. In a world of masks, COVID danced with science's grace. Nobel whispers hope. Alright, perhaps we should leave the poetry to the poets, and the chemistry, well, to the chemists. With that said, welcome to our video. Today we will talk about the Nobel Prize in Medicine, which was awarded to Adèle and Drew Weissman for their contribution toward mRNA vaccines. Let's encode their groundbreaking work together, if you catch my drift. Let's discuss why we need vaccines in the first place. The life cycle of a virus is a complex, multi-step process that plays a crucial role in the development of vaccines. Understanding this cycle is essential for designing effective strategies to combat viral infections. Here's a brief overview. First step in the viral life cycle is invasion and attachment. A virus begins its journey by identifying and attaching to a susceptible host cell. This attachment is facilitated by the virus's surface proteins, which precisely match specific receptors on the cell's surface. Think of it as a lock and key mechanism, where the virus and the host cell perfectly fit together. Once attached, the virus enters the host cell. This penetration can occur through direct fusion with the cell membrane or endocytosis, a process where the cell engulfs the virus. Inside the host cell, the viral coating is removed, exposing its genetic material. This uncoating is crucial for the next phase of the cycle. With the viral genetic material, DNA or RNA, now inside the host cell, the virus hijacks the cell's machinery to replicate its genetic material and synthesize viral proteins. This stage is like a covert operation where the virus discreetly uses the host cell to make copies of itself. New viral particles are assembled from the replicated genetic material and synthesized proteins. Once assembly is complete, these new viruses are ready to leave the host cell. They exit either by bursting the cell open, called lysis, or by biting off the cell membrane, a less destructive method that allows the host cell to survive longer. While the viral cycle is a marvel of nature's engineering, it presents challenges in vaccine development. The high mutation rates of viruses, especially RNA viruses, can lead to new strains that evade existing vaccines. Moreover, the ability of viruses to integrate into host genomes or remain dormant adds another layer of complexity. Hence, development of vaccine requires not mimicking the life cycle, but also staying ahead of these ever-evolving pathogens. Vaccines prior to the pandemic were designed to mimic the nature of the virus's life cycle that we just described. Step 1 would be identifying and sequencing the genetic code of viral code proteins and cloning them in a vector. Step 2 would be to place the clone vectors in our cells, and there our little cellular factory produces the proteins and ships them out of the cell, exporting them past the membrane. These proteins, once in the extracellular environment, are swiftly recognized as pathogens, and our cells rapidly produce antibodies that shield us, annihilating and resisting any viral offenses. This process is great and all, but it does come with its challenges. The main bottleneck here is that it requires a large amount of cellular cultures to be grown, making the whole process highly time and resource intensive. This is where mRNA comes to the rescue. To understand this, let us explain what is known as the central dogma in molecular biology. The central dogma explains that genetic information flows from DNA to RNA and eventually to proteins. mRNA plays a crucial role in the central dogma as it serves as a messenger, telling the cell what amino acids to recruit for protein synthesis. At its core, mRNA is a molecule made up of 4 different bases: adenine, cytosine, uracil, and guanine. These bases are super important as they serve as a template for tRNA to bind to. Each 3 bases in a row make up what is known as a codon, and different combinations of codons encode for different amino acids, telling the cell exactly what to build. Now usually, mRNA is produced in vivo by the cell, but around the 80s, scientists would leave the disco dance floor, head into the lab, and discover that mRNA can be produced outside the cell, known as in vitro transcription. This is a vital step in mRNA vaccines as it eliminates the bottleneck we described earlier, making the process much faster and more efficient as whole cells are not needed for development. Despite this, new challenges arose, the first being that delivering mRNA to the cells proved difficult, and new carrier liposomes had to be developed to allow their transportation. Secondly, the in vitro transcribed mRNA would bring rise to inflammatory responses, which were highly undesirable. This is where the findings of Adèle and Drew Weissman come into play. They found that you should like and subscribe to our channel. Jokes aside, but please do like and subscribe, they discovered that dendritic cells recognize and interact with in vitro transcribed mRNA. This makes sense, as dendritic cells are a type of immune cell that performs surveillance on the cellular environment, detecting any pathogens that come their way. The mRNA being made in vitro is unfamiliar to the cells, causing them to release inflammatory signaling molecules which can communicate with T cells. This is a strange result, as mRNA produced by mammalian cells does not cause such responses. But here's the catch. Mammalian-produced mRNA is usually base modified, so Carrico and Weissman hypothesized that they could perform base modification in vitro and avoid the immune responses. This is exactly what they found, which in the world of therapeutics and vaccines is very important. Why, you may ask? Well, think about getting stung by a bee, or eating peanut butter if you're allergic to them. Your body responds extremely aggressively, trying to kill the foreign molecules it is detecting, but at the same time, it threatens your life. If every time we took vaccines, we swole up, got red, and couldn't breathe, pharma would not be in business, which is a lose-lose situation. We would keep getting sick from viruses, and pharma companies won't be able to tear holes in our pockets. So Carrico and Weissman found that by modifying the base pairs of mRNA in vitro, they avoid the immune response and additionally achieve higher levels of protein production, all which are vital for medical applications. Imagine a world where our cells become bespoke medicine factories, crafting precise weapons against some of the most daunting diseases. This isn't science fiction, it's the reality of mRNA technology today. What if I told you that you might have experienced an mRNA application firsthand? With the COVID-19 pandemic, there was an urgent global demand to develop a vaccine. As we discussed already, mRNA vaccines can quickly be developed, and so this is one of the ways in which COVID-19 vaccines were made. Kinda cool to think that we got a shot of mRNA and that our cells know what to do with it without us telling it to. Secondly, cancer treatment. mRNA vaccines are the new frontier. Like tailors crafting a custom suit, these vaccines are designed specifically for each patient's cancer, targeting the unique mutations of their tumors. Once injected, our cells read the mRNA's instruction and produce cancer-specific antigens. Our immune systems, now savvy to these rogue cells, launches a targeted attack. Clinical trials are showing promising results, especially against formidable foes like melanoma. But that's not all. mRNA technology is also revolutionizing the production of therapeutic proteins. Picture this. Instead of taking drugs from the drugstore synthesized in a lab, our cells get mRNA blueprints to manufacture vital proteins right where they're needed. From correcting genetic disorders like cystic fibrosis to fighting inflammatory diseases, mRNA is like a versatile molecular architect, designing solutions from within. Welcome to the end of the video. Thank you very much for sticking around until the end. We'd love to hear your thoughts about mRNA vaccines, and please be sure to like, share, and subscribe so that you can stay in touch with our content. See you guys in the next one. Bye bye.